or such a huge problem and how they are going about and what is being done. Now, in between, I'll be asking questions and I want you all to put the answers in the question box. So don't just go off to sleep after logging in. It's your loss, not mine. Okay, so today we are going to talk about the trends of the emerging and re-emerging infectious diseases along with their definition, the factors which contribute to this, take a few examples as case studies like we did last time, and assess the public health response and the control. And again, a picture talk for you. Just see this. This is a continuation of the lecture last time. So when we were talking about the zoonotic diseases and we talked about the animal-human interface, look at this picture. This is so beautiful. This comes from the Australian Society of Tropical Medicine, and it clearly shows the kind of close interactions certain regions of the world have with the animals. So you can then imagine how easy it was for Ebola and Marburg to spread when the association was so close. So if you look at the trends of the infectious diseases have look at the trends which were there in the Western countries, the sanitization, housing, personal hygiene, antiseptic vaccinations, all these things, along with the development of antibiotics in the post-World War era, tend to make us get into a mode where we thought that we are over the infectious disease. We will no more, we will have infections anymore. But very soon came new infections, very soon came the older infections in a dangerous form, and then it became another problem. And then we started again searching for problems which could, with solutions which could solve these new problems. So, so the last quarter of 20th century, we saw that there were a lot of new and resurgent infections. We saw a lot of new and resurgent infections, and we saw a large number of new diseases like the rotaviruses, cryptosporidium, and then in the 80s came HIV and AIDS. So when we were in your stage, we used to think the only new emerging disease is HIV and AIDS because it was discovered in the 80s, and then we thought in the 90s everybody came to know about it. So we thought this is the end of the story and we'll not have any more. And then came hantaviruses, then came Lyme's disease, hepatitis C, E, G, and then came the zoonotic diseases, which have kind of, put us into a spin for the past 1.5 years. Now, if you look at the world which is moving, we have been able to bring down the infections caused by many diseases, and we've been able to eradicate a few diseases, some from some pockets and some from the world. So guinea worm and smallpox are the two diseases which have been eradicated from the world. Now, while they were easy to eradicate, should I say, or why they were, what were the reasons which led to eradication of this? Put your thoughts in the question box and I'll take them one by one. And let's see how the young mind thinks. Postgraduates as well as undergraduates. So just put your thoughts as to why it was simpler to eradicate guinea worm and smallpox and why we've not achieved the same results with, say, polio or TB or malaria. Then certain pockets had only diseases like yours and neonatal tetanus. Places where neonatal tetanus was where there were home deliveries and where they were using iron blades to cut the umbilical cord. So it was easy to eradicate them. Leprosy almost eradicated from the translation. Poliomyelitis, only 5 countries report cases of poliomyelitis now, so almost gone. But still the focus remains light remains that yes, at any point of time, they can get into a problem. But at the same time, when we thought that probably we are moving away from infectious diseases, came up all these new diseases, HIV, avian influenza, Ebola, Marburg. Many of these may sound like new names to you. And came the biggest resurgence, which is antimicrobial resistance. And just wait a few more years where it will be the only disease probably which is lurking. Then the emerging infectious diseases, how do you define them? These are the diseases which are newly identified. For example, SARS-CoV-2. Or it was completely unknown previously and they cause public health problems either locally or internationally. If they cause locally, then they are in an epidemic or form, otherwise internationally. And usually the time limit when we take for these diseases is about 20 years. Although it is not put in the definition, but we take as the emerging infectious diseases are the new diseases which happen in the past 20 years. So that's a fairly good time, by it's happening. Then we come to the story. What are re-emerging? Re-emerging are those which we've known for some time, but had fallen to a lower level where we thought that they are no longer a problem. For example, tuberculosis in the West was thought to have been, confined only to certain pockets of the poor and the slum dwellers and the black communities, and then or to developing countries like India or African countries and Asian countries. And they thought that we've conquered. Later on, when HIV epidemic came and the body resistance fell, tuberculosis came back with a bang in these patients. So tuberculosis so kind of re-emerged. Then in many areas where dengue was not seen, but in certain pockets of the world it was seen, that dengue re-emerged because those pockets, the rainfall changed, the climate changed, and these, the infections came. Then multi-drug resistant salmonella. Salmonella was believed to be, treatable. You give initially, people used to give chloroquine, then they shifted to fluoroquinolones, and then they thought, okay, we've done with it. Salmonellosis cannot kill any people. But slowly and slowly, salmonella started becoming resistant to everything, and then typhoid fever again became problem. So infectious agents which have been known for some time but have fallen to a very low level are showing an upward trend. That is what the prevalence looks like. Now let's just see what everybody's thoughts are on uh, smallpox. Okay, vaccination, herd immunity, rapid vaccination, diseases were having no zoonotic reserve, mass vaccination, no endemicity. Smallpox and guinea worm. Guinea worm was confined to a particular area, but it was not, smallpox was not low endemicity. Secondly, it was a very, very transmissible disease. Its R naught is very high. Then a known vector. Yes, known vector, immunization available, mass vaccination, human definitive. Then less mutations, stable structure. सब Google कर रहे हो. Smallpox transmits only from humans and humans. Disease like smallpox had a difference. So one of the major things which was there in favor of smallpox eradication was that you had a vaccination which was 100% effective. Now we all have seen how vaccines develop and how we go on learning about vaccination as the world goes on vaccinating people. So it was not an easy task to vaccinate everybody on this earth at that point of time. But people like us who were born before, I think 86 were all vaccinated. Then later on, the vaccination for smallpox stopped because the disease continued to not exist in the human population. There was no animal reservoir. For diseases, some diseases we still don't understand where the animal reservoir is. So it becomes difficult for these diseases to be eradicated. Now, why in the past few years we have seen that RNA viruses are emerging as a threat? Now, if you look at, they will have 4 levels of transmission. Now, this why I am covering is here is that in everybody's mind, it is SARS-CoV-2 right now. So if there is a level 1 where there is an exposure, now, this probably occurred in a stage before December 2019 when we talk about SARS-CoV-2. So this was a stage where it was exposure. Then maybe some exposure and some infection at level 2 occurred when uh, the infection spread from the wet Wuhan market to its users, employees, people, customers. And then it started transmitting. They all went back to their homes once the Wuhan market was shut down. And then they again started transmitting the same disease in their neighborhood. Because, see, at that time, nobody knew what's happening. So, what are the disease dynamic? What is the reservoir? What how this pans out? Nobody knew. So then they started spreading in their communities and then to cities. And then it because of the Chinese New Year, people travel a lot. They get 40 days holiday, so they travel a lot. So they travel to Italy, to Europe, and that's how the infection went from China to these places. So we they're very fond of traveling to Europe. So when they traveled to Europe, the infection went there. And then from an epidemic, it became a pandemic. Now, the same thing, if you realize, there are many, many viruses which still exist in these levels. So if you look at the level 2, these are very few viruses. They're Chandipura virus. The green ones are usually the ones which have been reported from India. So if you look at the Chandipura virus, it was from I think place in Orissa or Andhra. Now it remained confined to that place. It was easy to control and it didn't spread because probably the vector was not there. And so similarly, Casanova forest disease did not go beyond Karnataka. Similarly, Crimean Congo hemorrhagic fever. Japanese encephalitis happens every year in parts of Bihar and western eastern UP, but it doesn't spread to other parts of the country as much. Then came the level 2 viruses, which are like chikungunya, Crimean Congo hemorrhagic fever, Long, Venezuelan equine, West Nile. These are the ones which are common and these are the ones which are there, which have been existing. For example, rabies, rabies, Nipah. These are there at all level. These are level 3. They are there in the transmission stage. Then comes your level 4, which are coronavirus. It's a high level of epidemic potential. Dengue, hepatitis A, C, E, gastroviruses, enterovirus A and B, HIV 1 and 2. These are all in the epidemic or pandemic stage that they are created. So, what are the factors which actually contribute to this emergence? Why does this happen and why it is difficult? Now, if you look at our triad, you have an agent, a host, and an environment. Everything is interlinked. So, at the agent level, there will be an evolution of pathogenic infectious agents. That means the microbe is attacked. Wonderful example is your various variants of concern or VOCs of SARS-CoV-2 that you see. What is that? It is microbial adaptation. There was no change in the virus till the vaccine came. And suddenly the virus started mutating so fast so as to survive. Usually when a virus mutates, specifically an organism, mutates for the better. It mutates for transmissibility but it stops or reduces its killing. A virus which is very sturdy like a DNA virus, like the smallpox virus or the rabies virus or any other DNA virus or a very strong RNA virus also, doesn't change that much. And that is why it's very easy to control these things. So how they evolve is also very easy. Then they develop resistance to drugs, as I gave you the example of salmonella. Multi-drug resistant tuberculosis. Then they develop drugs to contain tuberculosis. We use them indiscriminately and then came multi-drug resistance. So it evolves, it mutates its own genes to develop and handle those antimicrobial things. Then the vectors started becoming resistant to pesticides and this was responsible for the chikungunya outbreak which happened in the Philippines a few years ago and then spilled over to India. Then factors we did them in zoonotics for a very long time. Now, demographic change. I gave you the example of Chagas disease and Ebola, how man went into the forest and started cultivating that land, It displaced a lot of things. You can see these larger animals would run to the deeper side of the jungle, but how do you displace the ticks and the fleas and this? They are all going to find new host, and that is the cattle of man and the man itself. So these demographic changes bring about a lot of problem onto the host. Then the human behavior, as the problem of sexual promiscuity increase, it is taken uh, very lightly that you have to be faithful to your partner these days by many communities. Earlier it was not so. So the way we are changing is also affecting us, and that is what people which had a different kind of sexual orientations came out in the open in the 80s and 90s, and that was one of the factors why HIV could be detected. Then the IV drug usage, sharing of needles, it led to diseases which were emerging of those pockets in certain parts of our country as well as in the world where IV drug usage is high, and that's where the prevalence of HIV, HBV, HCV is also. When the human susceptibility to infections are ideas of taking a protein out of a box rather than eat two eggs. Our idea of having exotic meat preparations which may be containing a lot of preservatives, containing our food habits, our behaviors can all lead to immunosuppressions. When you study for 16 hours during your exams, you don't pay attention right now, but then to study for 16 hours or 18 hours in a day, all you do is you suppress your immunity. And what happens is on the day of the exam, either you have not slept, so your mind is in a haze and a brain fog, or you are immunosuppressed and you end up with a cold. How many of you agree with me that you end up with cold on the day of exam because of your self-induced immunosuppression? Especially going out for our exam going postgraduates whose dead sheet has come by the way, and 12th of July is your first paper. So another immunosuppression I am creating for you today. So what happens is that this immunosuppression, you are in the hostel, you don't like hostel food, so what do? You junk, you survive on junk. And that junk puts you in the risk category of more towards tilts your food balance more towards saturated fatty acids rather than protein. And if the proteins are not there, what happens? Your immunoglobulins are reduced, your ability to respond to an infection reduced, and that is what brings about the problem. Then poverty and social inequality. Everywhere this is a problem. And only two people agree with me. My God. The rest of you are sleeping. Or Seka, you are too brilliant. You study whole day. You don't just study 16 hours in the exam. Okay. So environment, how does it help the organisms to give them the opportunity to come out from their niche to attack you? Because you see, your Earth has been developed in such a manner that there is a place for everybody. But if you go and disturb that place, if we want to explore even the Antarctic, and in that process, we end up killing so many seals and whales and penguins there. So what happens ultimately is that all that things come. Then economic development, land use, technology, industry, we've already talked about this. International travel and commerce. How did SARS spread? Not the SARS-CoV-2, mind you. SARS. We'll come to it later on. But just remember this, that within 24 hours, the infection had gone from Philippines to Canada. So you can imagine what role travel plays. Breakdown of public health measures, war, unrest, what is happening in Israel and Gaza Strip, and all that is going to take its toll later on. There are refugees, there are migrants, there are people who are forced to live in a one by 6 feet by 6 feet areas. So that ends up in creating a lot of health problems, spread of infections, and so. Then deterioration in the surveillance systems, there may be a lack of political will, there may be no political will at all, there may be no system at all. We don't know what's happening. All these factors, if you put together, the human growth, the population, the vectors, they're getting resistant, and then they put a pressure on the environment, and the environment puts the pressure back on them. Then we are using antibiotics like, sweet meat for animals, and that is causing the antibiotics to spill over in their meat as well as in the, in the food that is there. So that is another problem which is happening. Then you all put a lot of stress on intensive farming, year-round farming. So what happens is that we've read about zoonotic. So two-thirds of these infections with this, and if you look at the past 5 years, all emerging infections are zoonotic infections. And mostly it happens either because the animals have got displaced after deforestation or climate change, which happened with the case of Lhasa fever, or humans themselves go and penetrate itself. They come closer to animal reservoirs. That can happen in malaria. Plasmodium knowledii, which is actually causes monkey malaria, is known to cause infections in man because people like to go into the forest and they like to have some fun there. So that is what is. And then they are doing their farming there. They are using the jungle produce to bring wild berries to this thing. How many of you watch MasterChef Australia, by the way? If you ever watch that, they pay a lot of stress on indigenously sourced ingredients. Now, what is an indigenously sourced ingredient? What nonsense. They are going into the indigenous areas and getting produce from there. So that is that the right way? Now, this is what you remember I was talking about as to how, and you all had brought out all the PGs and undergraduates together had brought out all the project points related to climate and environment, and I had told you I'll show you something in my next class as to how global warming is responsible for the spread of malaria, dengue, leishmaniasis, filariasis, and so on and so forth. And this is the permafrost which I was talking to you about. What what has happened is that they found a giant virus. It was so giant that you could see it under light microscope. It's such a huge virus, and fortunately, they said that it's a commensal virus which actually swims in water and doesn't cause any disease, but was probably part of the reindeers which lived for many years. But then the permafrost melted, and this was found proven. And I have a small YouTube video. I hope it connects for you to see how permafrost melting takes place. Okay, so if you can see this, can you all see my screen? Whether this is playing? Can you see the video call button? All of you? Okay. I'll do this way. Even if can you all hear it also? Or should I give you the audio? There is no audio. I guessed it because the audio is coming in my ears and not in yours. All right. So I'll talk about it as we go ahead. All right. This is about an outbreak which happened in the Siberian area. And in Siberia, they found that after 70 years, they had an outbreak of anthrax. So they looked underground to see how this anthrax came about all of a sudden after 70 years. So When you see this layer thaws, but it is this layer which is always solid, and this is what is called the permafrost. It never melts. And the permafrost is limited to this area. So this is acting like a -80 refrigerator. So anybody at -18, nothing is going to die. So what will happen is that they will die and they get buried in this permafrost and will remain for 2000 years. This permafrost has remained like this. But as the global warming is happening, this permafrost thaws. And when it thaws, and we believe that by 2100, that is the next 80 years, only this portion will be left. Earlier was so. This is what happens. So this permafrost and and ground collapses, and this has happened in Siberia, Alaska, and these frozen animals then come out. So many animals have been found, and this material which is there decomposes and it will release these greenhouse gases, and it also releases the aerosol of the organisms which were trapped. So in Siberia, all these dead carcasses were responsible for releasing these anthrax spores. Anthrax spores will not die at -80 degrees also. So these anthrax spores were released and it killed huge number of animals. So all these diseases might be there in these animals and it's going to create a lot of problems. So now you realize why permafrost, and we talked about these materials as to why it's going to create more problems. So are we back to my PPT now? Why understanding this was very, very important because it immediately tells you that, if you believe that you've eradicated smallpox, but if the smallpox has remained buried in this permafrost, as it goes on melting, it's going to bring about, and maybe 40, 50, 60 years later, people will say, oh, it's an exotic disease which is coming and God knows from where it is. So the weakening of health structure, poor populations, they act as major reservoirs. Then there is a malnutrition and there is a severe infectious disease cycle, lack of funding in these areas, poor prioritization of health funds, and everything goes on in a vicious cycle. Then uncontrolled urbanization. This is something which we have known for the past. Right now, when you are in your 20s, try to take pictures of the border areas of this city and compare this when you reach my stage 20 years into your post MD and try to compare those photographs. You will be amazed. Unfortunately, people of my generation did not have access to cameras so easily. We used to have those film cameras. So I can't compare. I have to depend on someone else. If you do that, go to your neighborhood, especially those who are not from Delhi. Select those pictures and then keep it with you. And after 5 years, 6 years, after 10 years, after 15 years, compare them with the new photos and you'll understand how we are doing. Then the refugees and displaced people, so many other diseases which are coming because of all these human behavior changes in this. We've already discussed. So what are the examples of recent emerging diseases? Recent emerging diseases are plenty. Now, the ones in red have become a huge problem. The ones in blue are an endemic level, but they tend to remain there because of problem either in a particular area, but they cause, these are both emerging as well as re-emerging. So they cause a problem not only in one area, they may cause a problem in the other area. For example, typhoid fever may not be known in US because their food and water hygiene is much better than ours. They do not have a tropical climate as ours. But the same typhoid fever via a traveler can reach this place. Similarly, the hemorrhagic fevers which were there in this had those health authorities in WHO not acted so aggressively, they would have traveled all across the world. So what all we've had in the past, we've had the emerging and very emerging. This list will be there in any standard textbook, but what I want you to understand is that if you go from, as I said, we go only 20 years for emerging. If you go beyond that, if you go a huge list and we cover almost everything which you see now. There are some things which you see that, uh, and I'm going to talk about them also. The Nipah, SARS, the avian influenza, again came back in 2009 as swine flu or human influenza, and the avian influenza H5N1 modified itself and started killing man in 2006. Then Crimean Congo hemorrhagic fever reported from Gujarat, Ahmedabad to be sure. Then in MERS reported from Saudi Arabia. Then 2015 Zika reported from South America. And 2020 we all 2019 it should be actually not. Then in the re-emerging we have the various hemorrhagic fevers, includes dengue as well, chikungunya, yellow fever, Chandipura, West Nile. These why these are there are a few which will be here as well as here is because they came into areas where they were not known earlier. So that is emerging. Interestingly, we never find yellow fever in India. So find out why we don't find yellow fever in India. Then came the bacterial infections. Now these bacterial infections which you see in Elizabeth, Skinga, meningoseptica, New Ehrlichia, Wadley, these are all getting recognized because we have better identification systems. So it's not that these are emerging, but yes, as people are getting more immunosuppressed, these are making a huge impact. Immunosuppression is not just because of HIV, as that's in my next class, but just to give you a food for thought, there are many, many things which can lead to immunosuppression. The transplants, the medicines, the steroids. So all these mucormycosis after SARS-CoV is a classic example. Then emerging parasitic diseases, cryptosporidium, cycloisospora, uh, cyto, it should be cyto. Acanthamoeba keratitis and acrostoma. Now people are becoming more and more, uh, what should I say? They are becoming more adventurous and they go out swimming in these areas and that and that. It is leading to more and more organisms which are coming. So there are various emerging zoonoses as well, which we talked about last time also. Avian influenza, Nipah, Ebola, Marburg, hantavirus pulmonary syndrome. So you all see why they are emerging. So coming on to a few of them as to why it is happening. That is, first is MERS-CoV, that is viral respiratory disease which is caused by a novel coronavirus. It is called as the Middle East Respiratory Syndrome or the MERS-CoV and was detected in Saudi Arabia in 2012. And camels, which were very specific to that area in the world, were the major reservoir host. And that is why it was very easy to contain that disease there. And the civet acts, which are cats, which are often kept as pets by Saudi Arabian people, were the transmitters. So these two, because we could identify, we could control them and not allow them to spread. Then came SARS-CoV in 2002 and 3, and this came again from South China, just like the 19 SARS-CoV-2. And it was thought to be an animal virus, which probably came through bats and then spread to other animals like civets. And it was first infected human was found in the Guangdong province in 2002. And within few months, it had spread to 26 countries, resulting in more than 8,000 people. India was fortunate enough that we did not have cases of SARS-CoV at that time, although we were fully alert even then. And it was, but it has taught us a lot of things for which have helped us in the SARS-CoV-2 time. So if you see here, Hong Kong 1755, this is what I was talking about. If you look at the Hong Kong 1755, from Hong Kong, it went to Canada. And you see the maximum number of cases are there in Canada because after Hong Kong. So that was the power of travel. And that's what made us prepare for SARS-CoV-2, and there were lockdowns and there was no air movement in new nation states. Then come to our favorite COVID-19, which broke on December 31st, 2019 to call it COVID-19. Otherwise, it would have been called COVID 2020. So it was declared a pandemic in March. So it took a lot of time. 3 months it gave us before it became a pandemic. And it was SARS-CoV-2, which was very similar to SARS-CoV. And we learned a lot of lessons. We learned a lot of things which were there, and uh, that an infectious disease is a threat to all. I don't know why this animation is not playing, and you can see the whole thing here. The idea was to stop early. Then came in 2003, avian influenza H5N1 has been known in birds, and that's again a very important bird and man interface which is created. But it attaches. Why bird flu doesn't kill human beings? That is the xylem receptors which are present only in the birds. They're not present in humans, and that is why it was believed that will not affect the human at all. The majority of these infections were due to direct contact with birds infected with viruses, and we did get a few this thing. And the bird flu scare again came up in 2021 in January. This is from Times of India on January 10th, 2021, when there was an outbreak which was confirmed in Kerala, Rajasthan, Madhya Pradesh, Himachal Pradesh. Remember, there were crows dying in Delhi and Maharashtra. This is awaited, but then it was confirmed. So but it got contained very soon. Then came the 2019 swine influenza H1N1, which affected, you can see, these many countries. You can see the more darker red it is, the highest number of cases are there. So look at USA, Alaska, look at this, China, even India, and even like countries in South America. It was a huge problem. So it was a pandemic alert which was made to grade 5, and similar to what is right now for COVID-19. The problem here was the other day somebody was asking about the mixing vessel, and why pigs act as a mixing vessel here is that they tend to be receptive to all the strains of influenza. They are like the bats in coronaviruses, they are the pigs in influenza. So they get infected by fowl, they get infected by bird strains, they get infected by human strains, and everything gets mixed up in them. And they create a novel strain. So H1N1 will have some components of foul, some component of these. So it kind of acts like a chimera and creates a new virus. So that's how it happens. Then came Nipah viral encephalitis, which it was initially found in Malaysia, then Bangladesh, then Siliguri, and then southern Philippines. And in 18, last time we talked about, we had the Kerala outbreak, and the virus can be transmitted from bats to pigs. We discussed a lot last time about this. And I'm going to talk about, oh, this thing is got displaced. So this is about in May what happened to influenza, how it had spread like a pandemic. Now, what are the re-emerging diseases? Multi-drug resistant bacteria being one, and why it happens is I'm going to come to that in a short while. Then diphtheria. Suddenly in Eastern Europe and USSR in the early 90s, there was a huge outbreak of diphtheria, and why that happened was because of the disintegration of USSR, the health structure broke down. And that's what gave rise to this huge problem. Then human plague in 1994, after 15 or 30 years, reported from Shimla initially and then from other parts. 1994 was in Gujarat, sorry. It was reported from Shimla in 2000. Then dengue, DHF, because I can never forget 1994 because I was one of the people as an intern of this very college of Maulana Azad where I was posted on the railway stations to screen passengers as and when they came in by the trains which were coming from Gujarat, and you had to screen them for fever and ask them about history of hemoptysis and a scary scenario like we have now. And the only PPE that we got was mask. We didn't get anything else. And we used to get one mask a day. That's it. Then dengue for past 40 years there has been a 20-fold increase. chikungunya has also come and it's on and on. And now, why this animal antimicrobial drug resistance is happening is that not only is there an unrestricted availability, there is an unchecked dosage and improper diagnosis, and there is also an availability in the animal feed. So the anti-infectives which are used in plants and animals get concentrated in their meat, in their milk and other products, and it results in a loss. So farm food producing animals, in order that they do not become sick, they are giving antibiotics. And these antibiotics are given totally prophylactically and everybody uses their own dose. So they do this and this then in the manure goes into our food and then from here it travels to humans. From the animal produce it again travels to humans. And human beings are no smart, they also have so much of antibiotics and they all land up ultimately selective proliferation of antibiotic resistant strains and this is what we then another fact that we have to consider in emerging and re-emerging is bioterrorism. Bioterrorism is a deliberate attempt. So much of analysis going on what SARS-CoV-2 released deliberately with a lab strain. So it is quite possible that certain agencies release deliberately infectious organism and highest potential is anthrax, botulinum toxin, minerality, anemia, pestis, and there are sometimes diseases which are released only for destroying cattle because it can destroy your economy. The likeliest due to aerosol dissemination, just like a bomb. So that can be used. And then what are the control measures when we talk about emergency? So first and foremost is your surveillance. You need to have a very good surveillance system. You need to have very good surveillance systems both at the regional, national, as well as global level. So you have all kinds of surveillance. You should have like epidemiological surveillance, laboratory surveillance, ecological surveillance, animal surveillance. So the one health approach that we saw last time. Investigation and early control measures, implement these preventive measures like behavioral, political, and monitoring and evaluation. There is a global outbreak alert and response network, one which is governed and coordinated by WHO. Every Tuesday, even now, Wuhan releases an outbreak alert. Nowadays, it is mostly into which variants have been of SARS-CoV have been reported from you. Every Tuesday evening, they do release a global outbreak. It is a mechanism for combating international systems outbreak. So they ensure that this assistance is provided because it might be reported in an area where there are no healthcare facilities to diagnose a particular condition or to treat a particular, they may not have drugs to treat. So it also helps in capacity building and epidemic. So this is how the response sheets from various agencies looks like. Then what are the various solutions to this is besides the public health response systems in India, there is an integrated disease surveillance project or the IDSP, it is missing from here. The aim is to rapidly detect unusual, unexplained, or unexpected and track this information in real time. Nowadays, we have a lot of information, internet-based information technology. Even in our laboratories, we use a software called HUNET, and HUNET has got an ER system, epidemic or alert response surveillance, where you can even geotag that, okay, I'm in this village, and this village is reporting an unusual outbreak of food poisoning or water fever. So you can even geotag it and it gives out the world the coordinates of that project. Then the Human Genome Project has helped a lot. You know, December 31st, 19, the SARS-CoV outbreak was released to WHO in its epidemic alert by China. And January 11th, they had the genome uploaded on the genomic website, and that is how they could make the RT-PCR kits and the tests and other things so fast. Microbial genetics methods for disease control. We all know how variants are being tracked in genomic surveillance and so on. So we need to have a synergy between global, regional, and national. And we need to have a system where we have collection, verification, distribution, and response. Any failure anywhere, any break in this chain, and we end up into what we've ended up in the past 1.5 years. And this is the biggest example of the emerging diseases, of the zoonotic diseases, of the everything which is there. Now, last time I gave you a few questions. What is the response on that? How many of you have actually thought over it and submitted something? Most of you are saying that yellow fever vector is not found in India. What is the yellow fever vector? Find out and then let me know. The vector is also there, the agent is also there, the population host is also there. Why still the disease is not there? So, wow, what is the most likely cause here and why we've not seen this disease in India? 34-year-old male delivered by cesarean section and the child had microcephaly with calcifications. She has returned from Brazil and developed fever and rash and joint pain in the first trimester, but there are no active infections were detected against toxo, rubella, CMV, herpes. So her whole torch panel was negative. Syphilis was negative. Chikungunya, dengue, HIV was negative, and there is no other significant medical history. So what do you think it is? What is the most likely cause? No, it is not Ebola. Yellow fever. Just because I was talking about yellow fever, does yellow fever cause microcephaly in children? See, I can't cover all the emerging and re-emerging. That's what I have to read. Two people have said, three people have said Zika. Yes, it is Zika because it causes microcephaly in children. Why have you not seen Zika in India? What is the vector for Zika? Achha, let's go to case number 2. Uh, an Italian health worker came back to Morocco after a long stay in Sierra Leone where he worked for an NGO. The next day he develops headache, weakness, muscle pain, fever and keeps on deteriorating. And a picture which is very similar to what we see now, but the BP is 90 by 60, heart rate is 87, saturation is 92, and it has to be admitted to ICU. So what do you think the second one is? Ebola. Ebola. Okay. Do we see this in India? Have we seen Ebola in India? See, now here there are two things. Zika is spread by a vector. The particular species probably we do not have in India or is not breeding actively. So even if we got one or two associated cases, they did not spread. Now in Ebola, again, we did not have any documented evidence because the infection is mainly through your body fluids and it's a healthcare worker. So it's through blood and body fluid contact and all. So unless there was what happened with female Congo hemorrhagic fever also. It was only reported in a particular area and then that area was quarantined and the healthcare workers got infected who were taking care of them. So these things are taken care of if you are to take care of them locally. Now last time also I had given you uh, a thought which you have not sent, none of you have sent, to me at least. Somebody is writing Ebola was reported in Kerala. These are all travel related infections. I only showed you on the map how many cases were reported of few diseases like even SARS 1 was reported from Kerala. And they are usually travel related. You realize every time there is a pandemic or there is an outbreak outside India, Kerala is the first state to report disease. Why? Why does that happen? Any idea? Probably they are more vigilant. Migrant workers, returnees from Gulf countries, travel, all of them put together, but they are much, much more vigilant. And the biggest reason is they have a huge coastline and they depend on the coastline a lot for their economy. So they are very vigilant and they have quite a good testing tools in laboratories. Their own laboratories are linked. So that is why you always end up, whatever comes to India comes to Kerala first. All right. So, is SARS a zoonosis or not? Where are we on this? Okay, now what you will do is that you will mail the exercise that I gave you in the last class to your respective batch resident in charge. So, in my batch, I think Devyani is there. See, one batch will send it to Devyani and then she will share a small, each of you residents will share a brief write up as to what these children have written. You will come and share it with me. All right. Now, any questions if you have, please put them in the question box. Otherwise, keep on avoiding the elevators and keep on avoiding the flu. But don't take a pole walls to jump. Stay at home. So, we have any questions? So, again, I'm reminding you the questions I gave you last time. You will write them, take a picture, and send that PDF or WhatsApp that PDF to your batch in charge of the resident. And the resident will then compile these reflections and then bring it up. Why is yellow fever not found in India despite the vector being there? Exactly, food for thought. Open up your community medicine textbook, Park and Park, which you have forgotten, but there is a reason why that subject starts from first year. So open that book and you will see why this is not happening. There are many hypotheses. The actual reason is not known. There are people who travel from the area. Anybody who is going to endemic areas has to receive a yellow fever vaccination. So if I decide today to travel to Kenya or the Gambia or these countries, I will require a yellow fever vaccination. In Delhi, it is done at two places right now in government setup. One is the Airport Health Authority, APHO, and the second one at RML Hospital. So you receive a vaccination and the vaccination certificate should be at least 10 days old. So that vaccination certificate you have to show at your immigration. Only then you are allowed to enter the country and come back from there. So one is it's very extensive. Quarantining is there. If you come back from there and you don't have a vaccination, then you are quarantined for 10 days. The other is that although the vector is there, but probably there is some people have always said that because our vector transmits dengue, it doesn't transmit yellow fever. So all kind of hypotheses is there. The main reason is that there is an extensive lookout for the disease and that is why it is not there. Okay, any other question related to this? Now, let me tell you something. The last class, that was the zoonotic infections and the emerging and re-emerging infections. They are given very sketchily even in the latest textbooks. So this material I have collected from here and there, and if you want, you write a mail to me. I will send you the PPT also. But please, 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 you will not gain anything in the PPT. It is how we describe, but then it makes an understanding for you. But it's a competency which is there for your CBME curriculum. So it has to be covered. It's a new addition to your curriculum. Earlier, we used to only cover the zoonotic aspects with that particular disease, not separate. Similarly, for emerging and re-emerging. It was a need to know kind of area. Now it is a must know area. Whatever is there in your competency is now must. You have to know it. If there are no questions, I'll log out. All right, great having you. See you. Oh, one thing I want, don't log out. One thing I wanted to show you all. Hang in there, hang in there. I want to show you your attendance, which has been shared with me by Dr. Preeti. Can you all see? Can you all see your attendance sheet? Yes, ma'am. तो आ गया